Okay, back to talk about some basic kidney function and nephron function. So, left of that slide, here we go. So, general overview of kidney cross-section, a couple of things to add to this and point out. Cortex is the outer portion, of course, and the medulla is the deeper portion. And the medulla forms, these sort of look like pyramids. They're referred to as renal pyramids. There's the base, and there's the apex, sort of laid out on its side. So, and there are blood vessels that go along here, arcuate arteries, then veins, and I'm not going to ask you to know. But these are renal pyramids. And between those are what are referred to as renal columns. So when you're just looking at the gross cross-section, those are the renal columns. I don't recall ever asking anybody about those, but that's what they're talking about. Then there is, so the nephrons are in here, which we'll look through, and they dribble urine out of the apex into these cavities, which are referred to as a calyx. And a calyx, somebody thought it looked like a flower cup, the petals, which is, in botanical terms, referred to as a calyx. So there are minor calyxes or calyces, which is plural. And then there are major calyxes or calyces that have several minor. Again, not a big deal to stress about. So I talk about the calyces of the kidneys, and all of those drain urine into the renal pelvis, which then goes off into the ureter. The capsule is shown. And they are showing some of the nephrons. We're going to spend a lot of time talking about the nephrons. But there a good part of the nephron is in the cortex. Some of it is in the renal medulla, and we'll take a look at that. So just an overview. Next slide, we go a little bit deeper and looking at the nephrons. So again, there's the cortex, and there's the medulla, and this is draining out into a calyx. So maybe I'll write that in here just for orientation. And that's draining off into the pelvis. So the nephrons that we're going to be looking at, there are arterioles, because this is where we are filtering the blood. But what this picture shows is not the blood vessel system. This is showing more the tubular system. But at any rate, the nephrons are in the, most of it is in the cortex. Some of it descends down into the medulla. So the nephron here, let's circle this nephron. This one is mostly in the cortex. So this is referred to as a cortical nephron. cortical, meaning most of it's in the cortex. And this is about, well, let's also say that little of loop descends into medulla, the renal medulla. And these cortical nephrons are about 80% of the nephrons in humans. About 80% in humans. Turns out that we're not terribly water conserving like some other animals. The other type of nephron, most of this nephron is close to the medulla. And these are referred to as juxtamedullary nephrons. Juxta meaning close to. The thing to note on these is that the loop descends deep into the medulla. Whereas the cortical nephron just barely dipped into it. So loop descends, let's say, descends deep into the medulla. So two types of nephrons, the cortical nephrons and the juxtamedullary nephrons. Okay, that's enough for that slide. The next one is sort of an overview of a nephron. The cortex and the medulla aren't shown, but all of the, well, let's just go through the parts first. So one thing let's talk about are the blood vessels first. And there is what's referred to as a glomerulus, and then there's this capsule. So there are these round structures, and they have an afferent arteriole feeding them, supplying them with blood. Afferent arteriole. And then we have the efferent arterioles leaving the glomerulus and the efferent arterioles then go off to the peritubular capillaries. So it's one big vascular network to peritubular capillaries. Okay, so that's the blood flow, and we'll talk about that more as we go along. But this is an introduction to the basic anatomy of a nephron. So the blood enters, and this is where filtration occurs, is in this glomerular capillary. I guess I'll add that also with a leader or an arrow. Glomerular capillary. This is all in the book. glomerular capillary, let's say site of filtration when we're filtering the blood. Okay, so then we have fluid that's filtered out of the blood, and at that point we'll call it filtrate, and then it flows through this rather tortuous tubular system. This is not blood now, this is filtrate. It was pressed out of the blood. It's close to plasma. No red blood cells, and it just flows through here. And the first part of this nephron leaving the Bowman's capsule, and this is the capsule that goes around it. They kind of have the arrow. It looks to me like they have the arrow of this leader line going into the glomerulus. It should stop right on the capsule, right on the covering. So that's the capsule, and then it just kind of continues off. And the tube is convoluted and tortuous, so it's referred to as the proximal tubule, but I sort of prefer referring to it as the proximal convoluted tubule, convoluted for twisted, and that abbreviates nicely to the PCT. So PCT equals the proximal, closest to the beginning, which is the glomerulus. Proximal convoluted for twisted. Somebody has a convoluted argument. It's a pretty twisted argument. Proximal convoluted tubule. It's easier to write too, PCT. And then after the proximal convoluted tubule, the filtrate then flows into the loop of Henle. And it goes down on one side, and that's the descending limb. It makes a hairpin bend at the bottom of the loop of, this is all the loop of Henle. And then it goes back up on the ascending limb. So that's just the path of the filtrate. And then it flows into the distal tubule, which is also twisted and kinked. So it's often referred to as the distal convoluted tubule, or DCT. So the DCT equals the distal convoluted tubule. Okay, and we'll talk about different functions along the way, and then the filtrate flows into this collecting duct. So this is all the collecting duct. And different parts of this nephron do different things. So that's the intro to the basic anatomy and parts of a nephron. The next slide shows the vascular side of it. So there's a couple of things here. It's basically the same picture that we had on the previous slide. So I'm going to back up. So on the next slide that we're looking at, what they've done, since of course in the kidneys the nephrons aren't laid out like on a flat sheet of paper, they're twisted up. And the next slide shows this part of the nephron basically just sort of flipped over onto the other side. So that's what we're looking at, but the pieces are still there. So here's the collecting duct again. I'll abbreviate. And one of these limbs is going down, one of them is going up. At this point, well, we could figure it out. Let's identify the afferent and efferent arterial. Take care of those first. So here is the afferent arterial. So blood is entering, goes into the glomerulus, which is a series of capillaries, and that's where filtration will occur. And then the blood leaves, and it goes out through the efferent arterial. And we said in an earlier slide that that efferent arterial then goes out and branches and supplies the peritubular capillaries. And these are the capillaries that you can see that are just wrapped around this nephron. Because keep in mind, what we're doing is we're filtering the blood. So we filtered glucose, we filtered sodium out of the blood, and all of that is now flowing through the tubules. And if we don't get the glucose out of the tubule, it's going to go into the ureter and off to the bladder, because that's where this is going. This is going into the renal pelvis at the end of the collecting duct. Renal pelvis, and that goes to the bladder and then the toilet. So if we don't want to lose our sodium and our glucose and our amino acids in the urine, we need to get them out of that tubule. So we're going to move them out of the tubule into the interstitial fluid, and then that's going to then flow back into the capillaries so that we can reclaim whatever we filtered that we want to reclaim. So that's the basic picture of this slide. So keep in mind that there are two sets of vessels. So there's this tubular system in sort of a gold tan color, including the Bowman's capsule, the proximal convoluted tubule, the loop of Henle, distal, and then the collecting duct. So that's all part of this tubular system. And then you also have the vascular system. Which includes the renal arteries, veins, and all their branches, and the afferent and the efferent arterial, the peritubular capillaries. And while we're on this, I should mention that the blood vessels, the capillaries that wrap around the loop of Henle, are referred to as the vasa recta. Around the loop of Henle. Okay, so that's probably enough for this picture, with one other thing that we're going to spend some time on, so let's point it out while we're here. And that is this juxtaglomerular apparatus. So it's right where this circle is, and I don't want to obliterate the whole thing with a giant pen, but it picks up this piece of the, what piece is this? Let's see, let's figure that out. So it's part of the loop of Henle, so it's either descending or ascending. It's either going down or coming up. Well, if we back up and look at the picture closely, here's the Bowman's capsule, back up around the glomerulus. And so this part that I'm tracing with the marker, I hope that shows up. This part that's going down is the descending limb. So this has to be the ascending limb. And that piece of the ascending limb, you know, what comes after the ascending limb is the distal convoluted tubule. So right where the loop is ending and the distal convoluted tubule begins, that section of the tubule passes between the afferent arteriole and the efferent arteriole. So remember this, and we will come back to it several times. So we'll look at that. And the next slide shows another picture of it. Okay, so here we're zeroing in on the renal corpuscle. And a renal corpuscle is just the glomerulus, these glomerular capillaries. Glomerular capillaries, I'll abbreviate capillaries as just caps. Glomerular caps plus the Bowman's capsule. Those two structures make up a renal corpuscle. Okay, so that's what we have here, is a renal corpuscle that we're looking at. So there's the afferent arteriole again, supplying blood, and the efferent arteriole. And what we're just looking at on that juxtaglomerular apparatus was a piece of the ascending limb of the loop of Henle, and or depending on how you're describing it, it's also sometimes just referred to as the DCT, the distal convoluted tubule, which is how I tend to write it, I think, just because it's simpler to write DCT than ascending limb of the loop of Henle. But it's the same area. So if you appreciate where in that tubular system this is, you're good to go. And we will come back and talk about this structure and the juxtaglomerular apparatus later. So let me see if I can wrap a sort of circle around this. Oops, my pen cut out. Good. Juxtaglomerular apparatus. Stay tuned. But enough for now. So, a couple other things to introduce. Well, here is our proximal tubule, or our PCT, proximal convoluted tubule, same thing. The Potasites, the next slide shows those. They are modified endothelial cells that wrap around the capillary. They assist with filtration. I'll mention that again a little bit later. And again, this is the site of filtration, where fluid is forced out of the capillaries and into the capsular space, and then that leaves as filtrate. The yellow isn't showing up well. Maybe I'll write it up here also. This is fluid that was forced out of the blood. And it is going to flow through the tubular system. It contains glucose, amino acids, everything else. We'll come back to that. The last thing that I want to point out on this slide, which is important, are the cells that are wrapped around the afferent arteriole. So these are modified smooth muscle cells. They go by a couple of different names. Modified smooth muscle cells. So first of all, they are smooth muscle cells. They can constrict. It's these guys. They're wrapped around the afferent arteriole. And they allow for vasoconstriction, vasodilation, regulating the amount of blood that enters the glomerular capillaries. So they can control vasoconstriction and dilation. Constriction / dilation. And they also release renin. So you'll want to remember that. We've already talked about RAS and how the kidneys release renin. Well, this is where the renin is being released from, are these cells. And these cells, these modified smooth muscle cells, are also referred to, so let's do AKA, also known as granular cells, or JG cells. And I like JG cells because it's easy to write. It's quick. But the JG stands for juxtaglomerular. Juxtaglomerular cells. So you should probably recognize them as either JG cells or granular cells, but they are essentially just modified smooth muscle cells. I tend to follow and use the terminology of JG cells. Okay, the next slide shows just an electron micrograph of close-up, very high magnification, of potassites. And it's just thrown in here sort of as FYI, general curiosity, won't be on the exam or the quiz. I'm not going to ask you to identify these. Oh, good. Excuse me. But the potassites are these fingers that wrap around the capillaries. And so if I'm still being filmed on the side, they look sort of like my fingers. And there are two potassites that are wrapping around a capillary. And the fingers of the potassites get together, sort of interdigitate, and they form these little filtration slits so that the filtrate is regulated during filtration. So, kind of an interesting picture, but let's move on. So, we're talking about basic kidney formation and producing urine. Urine's produced in 3 basic steps, and we will look at these in sequence as we go through kidney function. So the first step is filtration. And we'll spend a little bit of time going through some of the details of filtration. And that is non-selective, which means basically anything small gets pushed out of the blood. Big things do not. So cells are not filtered out. Not cells. Otherwise, everything's small, including glucose, amino acids, ions, all that stuff is small. Could be antibiotics, it could be drugs, everything gets filtered out, the good with the bad. And then the next step is to come along and do selective reabsorption. And this is selective. So it's a little bit like the old, the old saying, throwing the baby out with the bathwater. In filtration, you're throwing the baby and the bathwater out the door. And then the second step, you go, oops, let's go back and get the baby and everything else that we accidentally tossed out and bring it back into the house or bring it back into the blood. That's kind of the idea. And it requires specific transporters to do that. And we have specific transporters for glucose, amino acids, etc. that we'll look at. But we don't have specific transporters for things like antibiotics or other wastes. If you don't have a transporter, it stays in the filtrate and heads to the bladder. And the last step that I don't do a lot with is excretion. No significance for changing pen color. I just had green going. And some examples, we can excrete hydrogen for pH and urea for nitrogen. So, last slide in this section is looking at glomerular filtration. So we're starting the first step, starting our discussion of filtration. And this is there's not a lot to it. We've already been through something like this when we were looking at capillary exchange. So the basic idea should be pretty familiar. So the diagram is representing a renal corpuscle, the afferent arteriole and the efferent arteriole. So blood entering, blood leaving, and filtration is occurring here. We're forcing fluid out of the blood by blood pressure or hydrostatic pressure. And so we're back to the old idea of forces out and forces in. So the main force pushing fluid out is blood pressure. And in this illustration, they've defined pressure as pH here, or P sub H, and the H is for hydrostatic pressure, but it's essentially blood pressure. So whatever you call it, P sub H or just blood pressure or hydrostatic pressure, it's all the same idea. It's basically blood pressure that's pushing the fluid out. And then you have a couple other forces drawing the blood back in. And we had the colloidal osmotic pressure. We looked at that before. That should be familiar to you. And we're again talking about capillary exchange. And that was determined by the proteins in the plasma. And the primary osmotic protein, do you remember what that one is? The albumins. Remember who made the albumins? Liver. So, same idea. We have osmotic pressure in the blood. And then we can also have some capsular pressure. So this diagram just defines that as P sub fluid, and that's basically the back pressure. It's the fluid that's in the capsule. So it's essentially hydrostatic pressure, HP, in the capsule, or whatever symbols you want to use. That's the idea. So the numbers that they've given here are blood pressure 55 out. So forces out would be 55. Everything's in millimeters of mercury. And then what are the forces in? Well, we have osmotic pressure. I'll just go back to osmotic pressure, OP, of the capillary or the glomerulus. And what else? We have the hydrostatic pressure. Let's not use capillary here because that's going to get confusing, or it's going to be confusing with capsule. So let's go osmotic pressure of the glomerulus. I'll go glom for glomerulus. And then we have the hydrostatic pressure in the capsule. Bowman's capsule. It's back pressure. So, pretty simple math. So, NFP, our net filtration pressure, is equal to 55 minus, we didn't put the numbers in here, minus 30. No, let's leave the minus sign off of that. Otherwise that will also get confusing. And the hydrostatic pressure was, I'm making a mess out of that, 15. So we have minus 30 plus 15. And that equals 55 minus 45. And our NFP equals 10 millimeters of mercury. So hopefully you're comfortable doing that. And I'm going to stop there. The next figure is a long one. It's a complicated drawing. It's one that I sort of drew by hand. I usually do that on the board. And so I'm going to stop for now, and we'll start. I'll make another lecture for just 2 slides. So see you soon.